I just, because I'm touching page. That's like a month later. 'Cause I saw it.
All right, guys, let's get started. Today's gonna be a busy day. I'm gonna give you guys a lot of stuff today. So try to get the big picture out. We're gonna be going back and forth to this stuff for the next like 2 weeks. So I'm gonna try to keep it on the surface, but I might dive in a few times, yeah? All right, so the big thing we just talked about before was, let's get the little crew drawing. We started on that here, right? We're bragging about, hey, how cool is that platinum member? Two big features we care about is hydrophilic on the outside, but it has this hydrophobic core. This means that we have this cool power where now we can control it, or it blocks things from coming in. It's like a little barrier. It'll help us be able to build up molecules in different places. It also gives us the ability to, like, we can actually pump them and put them in different places, and we can actually build them up without them diffusing out, right? So big, powerful barrier. The thing we're gonna be talking about today is, now that you have this like beautiful, basically shield, it's gonna block things from coming in, how are you gonna have two neighboring cells talk to each other? So the thing we're gonna be chasing down today is gonna be, how do we have these molecules that are gonna be working basically across this membrane, right? So on one side, we're gonna have things that have to be embedded in the membrane, so they can receive signal from one side and get it into the inside of the cell. In other cases, I'll talk about how there's some molecules that can go through the plasma membrane, and then we can actually have the molecules work on the inside. But the general strategy is gonna be that, if you imagine this to be a cell, right? It's gonna have a component for molecules that can't cross the membrane itself. They're gonna be seated and kind of stuck inside that membrane, right? They're gonna be waiting to get the ligand for that specific thing that's gonna come in to activate. We've worked with a few of these before. If you guys remember when we were doing the protein folding, right? Remember it was Anne's favorite, what was it called? The protein folding? Like IREI or something like that, right? That was one. I think the other one was called like ATK4 or ATS6, yes.
Perks.
What's the one?
Perk 6, I think. Something like that.
And remember, their thing was they were sitting on that membrane, kind of waiting, right? So that would be like a purple component. When your misfolded protein came in, which in our cases were the amino acid and hydrophobic regions, that would be our ligand. They would bind specifically to that pocket. That'd be that outside signal. We're gonna transfer that information to the inside of that membrane, right? Again, back to our protein example. Here we had things where some cases they acted like kinases, right? They would phosphorate each other. That would be like this internal part of the components. They would do a different one, but basically you need some information to be happening on the inside. In one case, it was the kinase function would set up, and then the nuclease, it would do splicing. And then eventually we'll get to this part where we have this functional molecule. In that case, it was a transcription factor. That thing could then go and do a function, or it can go into the nucleus and change gene expression. So we go from a signal comes from the outside, right? It binds to a specific receptor. We transfer that information to the inside of the cell, and we'll use different components on the inside to get us to this final thing of power and change genes. So as I go through today's lecture, I'm going to talk about a few of them. Over the next few weeks, I'm going to be hitting a few more. For these, I'm going to tell you exactly which is going to be my favorite. It has like there's 9 steps to how it passes the signal. So it's a beautiful midterm question, right? But the goal is going to be keep track of the molecules, and I kind of want you guys to think about like we're working our way down the cell line, which is you're going to have your ligand on the outside. It's going to transfer into a receptor. Then we have to have different components that get us all the way to the inside where we finally have the effect. Most likely it's going to be in the nucleus. Yeah. All right. But for this one, when you guys do your drawings, I'm going to show you a video. When you do your drawings, I'll share my notes with you guys for this case. But some of these molecules, they change conformations. So when the signal's on, they'll look one way. When the signal's off, they'll look in a different conformation. Be ready to prep how to draw both different cases as you do your story. Yeah. All right, let's get started. All right, cool. So this is my beautiful drawing. That's one. Let me get the big screen. So everyone's feeling pretty comfortable with this super general thing, right? Like we basically have 4 steps we need to work on, yeah. So now let's zoom into some of these factors. First, we're gonna talk about a little bit of how these can vary, right, your liking. So again, one of the cases we talked about was forward protein folding. That hydrophobic signal was that thing that's how they recognize that molecule, right? The signals can be anything. They can be gases, they can be lipids, and they can be proteins, right? So in that case, it's whatever molecule you make, whatever shape it has, it's made specifically to bind to a specific type of receptor. And that's how we know that one signal will only work on one of those pathways. On the next part of that is going to be, again, here I have it in black is that receptor. Over here would be like this purple part, which is, this is the one that's responsible for recognizing a specific partner. And in my head, I'm thinking like, they're like perfect handshakes to each other, right? Like, it's like, you know, you have your business handshake, you have your friends, you have hugs. That's how these basically work in pairs, right? If you try to shake someone's hand and they go for a hug, it's a weird, you know, it doesn't work. Just like that, ligands and receptors, they have to work as a team, right? The next part of that is when they're bringing that signal on the inside, I want you guys to be thinking on this inside part. You have that same type of like very specific type of handshake or interactions, right? The shape of one receptor will be properly shaped so it can do its handshake with the next intercellular component. They'll put specifically matches to each other, and normally it's going to be that when this changes shape from the outside, this modifies, and this can change the way the instrument function, phosphorylating and changing the way they can clamp onto each other, right? At that point, you basically have different cascades, and eventually getting us to this basic gene expression. And because we have so many partners that we can make, right? Specific molecule, specific receptor, specific pathway, we can control a lot of different mechanisms, right? I'm gonna talk about, I think, hormones today, just lightly. And I think I'll probably have a video about pharmacogenetics for bacteria. That one looks pretty cool. Let's go. All right, so again, bringing in my little rough drawing, we're getting into this context, right? So I said they can be very specific about what ligand and what receptor you use to kind of set off the signal. What you guys are going to then see is that you sometimes end up hitting different molecules as you go through it. This has 2 really cool features to it, right? When you're hitting the molecules, in some cases you can do it where it'll go down a specific chain, like one molecule activates the other. In other cases, you're going to see things like this, where one molecule itself can activate like multiple targets, and each one of those can then again hit multiple targets. So you can imagine this could be a thing like, I think the cool one in bacteria is like when they're sensing that they're running out of sugar in the environment and they need to switch for the metabolism, they have things like this where one molecule on the outside, knowing that they're low on it, is going to set up the whole system to like, let's turn off a whole library of genes, let's turn on a whole different library of genes so we can work in the environment. So first big powerful tool, they can branch out. The other big one that they can do is that when you turn, let's say you have your receptor, normally this is on the off state, not doing its thing. At this level, you can have also thing called an amplification. So that ligand could be there, it activates the receptor, and then this arrow, it ends up being active for, let's say, like a minute. But during that minute, it's going to interact with more and more of these molecules. So technically, even though you only started with one ligand, down here it's going to be multiplied. However long it's going to be down, you'll make more and more copies of this. If this gets multiplied by 10, this can also be multiplied by 10. And as you can keep going to the thing, one single molecule gets turned into hundreds of copies of this guy. Right? And that means that even really small amounts of signal can actually have super huge outputs and controls. So it's a really cool tool. Like when we normally study the changes in the molecules, like you can have stuff go from non-existent to 10 to 15,000 copies, just because you can try to amplify itself through that state. All right. Okay. Let's go. All right. So part of this communication, there's different types of way that basically they can talk. So now I want you guys to kind of think like we're zooming out a little bit. We're not just thinking about a single cell. Like now we're thinking about here about how two different cells can interact with each other. In this case, the thing in green and in purple, when you have direct cell-cell signaling, it's again, it's like imagine on the cell, the handshake is some kind of code of like, hey, everything's fine. There's some cases where you can have two cells, they both have this type of setup. And then it's kind of like they can both receive the signal or transfer to each other, like how they can grip onto each other. In this case, you can imagine this receptor, it's gonna work with this protein. This will then be able to give us the signal, give us the nucleus to change the signal. This is the way that they can detect each other. They can also communicate different things. For example, that first cell on the left, if it stops expressing that green receptor, right, it's gonna stop being able to grab onto their cell. Other ways that we can talk is gonna be something like this, which would be, instead of both cells having to be directly grabbing onto each other through their membranes, you can have one of them make a molecule, and let that molecule basically diffuse till it eventually to the ligand, till it goes and hits the receptor in a different cell. Some of these you guys will hear about them where it's like, this could be local ones. I believe on my side note, this one talks about like, this is kind of what you see in our neurons, where you have them right next to each other. They produce the chemical glutamine. The receptor does so will take it right. You can also have these, some of the cells that have like positive feedback loops, and they can actually make the molecule themselves, release it to the cytoplasm, and it'll come activate themselves. That one's really cool. Not cool, it's bad, but it's one of the cases in cancer. Cancers put out a bunch of growth hormones for the cells to keep dividing faster. Then they detect the same hormone, they're like, oh, it's time for us to grow. Let's keep going. And super bad for cancer. But this is the local communication. We can also do the same thing with. Yes, sir.
Is there any situation in which autocrine signaling like wouldn't be harmful to us, or like there are situations where it's beneficial?
Oh, that's a good question. I feel like the one I'm getting off the top of my head right now, it's in, I think it's like for how you build up the hormones. Is it like during for ovulation? Those signals will signal the other cells to kind of produce more of the local signal to ramp themselves up. I think the other place too is like your immune system. It'll be like your cells that are normally going around looking to eat up all the like infectious agents. They'll ramp each other up with signals to be like, hey, there's a bad chemical here, or there's something that's attacking us. It activates the defense mechanism. When they put it out into their local environment, any other macrophage that shows up, it's going to get hit by it, but it can also hit itself with it. And that's when you have our stuff like our immune system. When we have allergies, normally we swell up. That's because all the cells are basically just going crazy at the point to be like, let's build up the amount of cells in that spot. So those local, now we're talking about the long distance. In this cases, we're going to bring up hormones. These end up being produced in one cell type. They're a lot more mobile, so they can actually go into our blood system. They can travel from our brain to like our different organs, right? At which point we have the same thing. They go, binds the receptor, and we can basically signal. For this one, everyone normally talks about these guys. It's talking about different hormones. So you guys know there's testosterone, estrogen, progesterone. So testosterone and estrogen are normally like if I start taking testosterone, I'll probably get buffered, right? I think estrogen is basically the equivalent for females. Then progesterone, I think, is to do with fertility, like for having babies, like at times the cycle. But structurally, they look pretty similar, right? Like you can see, you have these four ring structures. Later on, I'll show you how like they have these like slight modifications in functional groups, like the methyl group, the oxygen group. And that's just enough difference so that when we're talking about that ligand matches up with a partner, that'll make this bind with one receptor. This will work with a different receptor. And that's what gives us the ability to have two different signals from something that looks very similar. Oh, there you go. That's the cool picture. So here are going to be some of those hormones. Again, look at those four rings That's the general structure. This color code means nothing. I was just trying to separate, you know, what I thought looked different or similar across all the groups. Like you can see here, these three have the long O's. There we have a hydroxyl group. But that's the difference between, you know, if I start making that, I can get a lot buffer. I'll be like, let's go. I think this one's cortisol is for stress, right? So like if you give me this, then you just start making me feel like high anxiety. But the cool thing about these molecules is that they normally have a lot less polar groups. So right, these will be the ones that I have a little bit of trouble trying to get to the membrane. But actually they can still kind of diffuse to it because they have this big carbon structure. The cool thing, or the only slight difference with them is because they can come into the cell, we can basically skip this step, right? We don't need a receptor waiting for us to get things done. They can just come in themselves and they'll feed into basically the same kind of pathway. They'll hit some effectors, they'll change the way they function. These will go either grab onto the DNA differently, they can interact with proteins differently. So that'll be a different power. The one I used to work with was, I used to do a glucocorticoid receptor. So it detects sugar. And my trick was I would take it from rats, give the DNA to the plants, and that receptor, when it's not signaled, I just say this green thing, it sticks in the cytoplasm, right? And then what I would do is whenever I added the specific, like the hormone to get it activated, it would go bind the receptor, it would change the conformation, it would get released from its partner, it would move into the nucleus, and then I could just attach different tags and I could turn on genes whenever I wanted. So super powerful stuff. But again, it's just a little hack that we don't need to go through that membrane receptor. We can just come right in and we can hit those molecules. But technically, it's almost the same pathway or very similar techniques. All right. So now let's talk about, so the hormone's a little tangent, but let's come back over to the big picture. And let's start thinking again about those purple players, right? One sitting across the membrane, trying to catch the signal. This one, we're going to talk about 4 different classes. We're going to start with, so nuclear receptors, ligand, gated ion channels, G protein coupled receptors. Those are super famous. So I'm probably going to go a little bit more into that one. And the final one are going to be our enzymes. Some of the enzyme, it'll be like the UDP stuff we talked about earlier, but let's go into them. All right, nuclear receptors. This one kind of works a little bit like the one I was talking to you guys about. There's the receptor, and this is basically the hormone coming into it, right? It's normally trapped by a heat shock protein, and it will stay in the cytoplasm. The moment you add that hormone, it changes the conformation. These two things can interact anymore. This thing has tags on it that send it to the nucleus. It's able to come here, right? This allows it to use its DNA binding domains, right? Like this little DNA part of the DNA binding domain, find its target. It also changes the conformation, so now it can interact with more partners or different partners. Big player here is going to be the other proteins that are help it set up with activation. Yeah. So pretty cool thing. But again, we can basically bypass the receptor because we can come right in. These other ones are going to be our ligand-gated ion channels. So in this case, our signaling, our ligand, the green thing in my picture, would basically be here, this red molecule. Right? In this case, when the system is off, the molecule is not present, you're going to see that our basically this will be our channel is going to be closed off. Binding of our ligand to that orange receptor ends up changing the conformation, and that's just enough to make it so we can move ions in and out. This will be free diffusion, so they just basically go down their gradient. But then normally what happens is these ions, when they get inside, they'll basically change either the charge of our cells, or they can change the way that a molecule functions inside. And that'll be the way that we can actually transmit the information. Like for neurons, I think when you guys go into like the shape of a neuron, you normally have like it has a polarized charge, and then all of a sudden it's going to depolarize itself, and it'll do it through a mechanism just like this. Brings in all the ions, cancels out the charge. All right, next one. Ooh, all right. And his favorite is back. There they are, IRE1, PERT, and ATF6. Right. Again, these follow This first layer would be that purple layer, you have the receptor. Our ligand on the top of this is those misfolded proteins that have hydrophobic parts being shown on the outside of the protein. When they come in, this will be this first level of function, which is they act like kinase and they act like nuclease. They do the splicing and then they make that first transcription factor. That'll be the one at this level here. Right, transcription factor goes into the nucleus and we can change gene expression. The other ones do their effect slightly different. You guys remember this one? They act like kinases, they phosphorate each other, then they phosphorylate EIF2. By doing that, instead of working this way, right, they work a little bit going this way towards the function. Without EIF2, you're not making any more proteins. That's when that second molecule doesn't care about EIF2, it'll translate itself. That other molecule is the one that comes in and changes gene expression. But the strategy is technically the same. ATF6, close one, but it does it a little bit differently. Because in the first two cases, when the receptor is on or off, because it has those like alpha helixes that trap it in the plasma membrane, it can sense the signal, try to print something out, but it can't actually read that it's stuck there. That's why it has to use different molecules to get its information out. ATF6, that one has a pretty cool neat trick up its sleeve, right? So remember for that one is, once it's bound on the outer part, the ATF gets activated, that protein then leaves in a vesicle, shows up at the Golgi, it'll get cut. Once it gets cut, that inside portion gets released, and that is the actual transcription. Right. So in that case, in this rough drawing, it would be something like, once it gets activated, this thing splits up, and this equivalent would be this little shape is released. And this is the one that can directly go bind to the right. So it's a pretty cool trick. The cool benefits about that one is that because you have the same receptor and the molecule together, it's like the moment you activate it, you can for sure have your downstream signal to keep the signal going, right? Sometimes, like, for example, this guy, IRE1, it can bring the signal in, act like a ribonuclease, but if that RNA isn't present, it won't be able to splice anything, it's not going to change the signal. So it needs to make sure the other things are present. They're a little bit more finicky. That thing, because they're all together, it's like the moment you signal, you're for sure going to release the one. Some of the cells, they'll kind of use this signaling to be able to behave differently, and you could do it at different levels. Some of them will do it at the top by whether or not your cells have that receptor. The next level will be like these intercellular components. Each cell type could have different intercellular components present, and whether they're present or not, that's a good way for you to change your function. Yeah. All right. We got one more to go. G protein. Everyone's favorite. From now on, you're probably going to hear this in the next like 3 or 4 classes you go to. For like most molecular biology or any other class, they'll always bring these guys up. So for these, sorry, G protein. I gotta have myself. Yeah, the G protein. So for this one, it's basically G protein is composed of 3 different amino acid chains that kind of work together. So alpha, beta, and gamma. Right? In the off state, they sit as a group just like this, and they'll hold on to a GBP molecule. So that's a guanine with 2 phosphorus. Once it gets activated, what ends up happening is we switch out that GBP and you bring in a brand new fresh GTP. That ends up releasing the thing. You will have the activated G protein, and basically the beta gamma will get kicked off by themselves. Right? So if this is the off state. On the on state, we get something like this. B gamma, and then here you have your alpha. And then now this is basically on, and it'll be functioning. While this guy, when it's trapped together, is basically off. Right? Now that thing will be able to go work with a different receptor. In this case, it works with a denominating cyclase. it then activates this guy. This will then be able to, it removes two phosphate groups from the ATP, right? When it does so, the signaling pathway ends up producing the cyclic AMP. Cyclic AMP will then go and work with this next batch of receptors. Here you have a repressor component and a catalytic component, right? These R and C. In the off state, they're gonna be found like this with each other, completely bound up. So the catalytic is being held off by the repressor domains. Once they start sensing the cyclic AMP, it'll jump onto them. It'll make the repressor domains want to fall off and release the catalytic ones. And you have these basically empty purple ones. Yeah.
What's the GEF?
Oh.
On the third bullet?
Perfect. See, it's right now, I'm out of order. Let's go back. GEF and GAP, right? So when we're looking at jumping between this state and this state, we have here, this will be GTP, right? What we have attached to this guy. On this one, we currently have it as GDP. So for the first part, we have GEF, which is one nucleotide exchange factor. So what you're going to be doing is, this receptor is going to be here. It currently has the GDP. We're going to actually kick this one off. Like it'll get removed, and you bring in a brand new GTP and replace it. So that'll be this little part right here, right? This GDP got kicked off and it went out. Brand new GDP came in. Now we have this active alpha portion of the G protein. As it's doing its job,
That's only for the alpha.
it's only for the alpha.
Is that GTP ACE? Is alpha?
Yeah, so what's gonna happen is when they're separate, this is gonna hold on to the GTP. When it's time to turn this thing off, this will come back into it, and now it wants to act as a GTPase. GTPase ends up removing one of the phosphate groups. Right? And you can see now that the G protein has been built back up, GTPase function happens, the phosphate gets kicked off, and now we're back to that GDP, that off phase. You know? So by controlling this molecule, like the beta and gamma unit, in this case, this will let it behave as a GTP, and it'll have the alpha, we can have the signal on. When this becomes back on, then you turn on the GTPase function. You're gonna kill off the GDP, and you turn the signal off. Make sense? Yeah. All right. Let's go back to this guy. Yeah.
What's the part with camp?
What?
The camp protein after ATP and cycling.
This one, protease K? I'm sorry,
CAM.
CMP? All right, cool. My drawing's gonna help you guys out. I drew it nicely here. If I didn't draw here, that's on me. All right, so the drawing's not that good here, but ATP is just like basically one from DNA, right? You have the 3-phosphate and you have the adenine. The cyclic AMP, what happens is it drops off 2 of the phosphate groups, and then it's gonna make a link across itself. Let me show you a picture. So check this one out right here. So you see, before it had phosphate, that was your ATP. After that enzyme, you basically catalyze this reaction. And now it's basically looped back onto this 5' side. Oh, sorry, 5' side is coming into the 3' side. So it'll be like a single nucleotide, basically glued itself into itself. And that'll be the molecule that our cells normally use to tell them that we're kind of low on energy.
It's that one I just went down the slide.
All right. Where are we? Cyclic AMP. All right. Let's start off. I'm not gonna start from the beginning again one more time on this one. Again, I'm gonna share my notes with you guys, but I think they still need a little bit like a little paragraph to talk about. So if you guys let me draw this out, right? You have a receptor sitting at the top. That'll be our receptor, right, this first part. In the off state, nothing's gonna be bound, right? So this is for the epinephrine system, our epinephrine receptor is gonna be off. In the on state, right? We expect our receptor to have a ligand present, right? That means the signal's gonna be on. At the next level, now we're gonna move to the G protein. In the off state, right? The alpha, gamma, and beta, they're all stuck together, right? Everything's off. On the on state, your alpha, your beta, and your gamma, they'll be sitting as a group. And over here, you would have your alpha sitting with the GTP. Right? This is now a functional thing. This will keep the signal going forward. Right? At which point the GTP with the alpha will hit this adenylate cyclase. That'll activate the next part of the signal, right? So I'm gonna write it here as AC. Again, in the off state, it's not doing any interaction, it's not having any function. In the on state, this is helping us do that transition of you have your ATP on one side, right? And this is going to be being converted into that cyclic AMP. So in the off state, technically we have a big buildup of just ATP. Right? So high concentration of this, you don't have this molecule present. Right? Now having this molecule, when we get to the next step, that's the one that's going to go get this into release. Right? Cyclic AMP shows up, binds to the repressor domain, and we'll kick it off. So again, off state, repressor's present, and they're clamped on to your catalytic ones. These are ones that are going to be doing the function, but they're turned off. Down here, we're going to find our repressors. They're going to have cyclic AMP on them, but we're going to have the catalytic sites. These will be ready to keep sending the signal forward. Right? Once we do that, this goes to the next one. It acts as a kinase, gives a phosphorylation, and it activates another phosphorylase kinase. Right? This molecule gives us the phosphate group. So if I kept going, here would be that activated phosphokinase. And what it's going to end up doing is down here, you get a phosphorylation. The signal keeps moving forward. On the off state, because there's nothing to signal to it, our APK is not phosphorylated, aka on the off state. Does that make sense? Yeah? All right.
What are the green things on the red thing?
These are the cyclic AMPs.
Okay.
Like those little, they have them here as the white one. But it's the one that we brought up for the Paula brought up, right? It's the with the sugar ring.
And then what's C?
See?
It's like the protein in the middle.
So this one,
It's a
they're
protein.
called, this is the repressor, and this is the repressor domain and catalytic domain.
Okay.
Together as a group, it's called protein kinase A. So this will be like that blue purple region. Normally they always, like when you guys will see it in the text, they love to shorten it to PKA. And everyone kind of knows that we're talking about this protein kinase A. So here we have the repressor, and here we have the catalytic. And here this will be the catalytic, and these will be the repressors, right? Basically, they're free from each other now.
Okay. And once they're free, then it lets it go down the pathway more.
When it's free, now the catalytic thing isn't being repressed anymore. So now it's a catalytic. Or like it'll basically, now it'll be able to catalyze the kinase function.
Okay.
And that's what it'll be using that ATP, catalyze the reaction, and we give off that phosphate. Now we turn this guy, right? This one does the same strategy. It goes and it phosphorylates the next molecule. Now this is the one that gets us all the way down here. Right, we're right above the function. That'll go, Sorry, my bad, I'm mixing them. Here it'll basically switch from glycogen to this glucose 1-phosphate. For this, glycogen is basically something like this, just a bunch of sugars kind of linked into each other, right? So remember, your sugars should be your 6 carbon rings, right? Six carbon rings, and you've linked it up to another one. When they're bringing in that phosphate group, you're basically transferring it here, and it's gonna end up releasing it from the other molecule. So you'll have like the stored sugar in this huge network. At the end, that phosphate group that's down here is gonna get transferred all the way to right here, and this sugar will be free by itself. And you just put glucose into your system. Yeah. So for this one, again, I'll share my notes with you. They're a little bit messy. I hope that you guys can sharpen them up a little bit, work with them. The really cool things about this one is, when you start off with this one, epinephrine, that very first line all the way at the top left, that's the signal they sent to that cell. Right? When it has the receptor and it activates the first part, it's still a 1-to-1 reaction. Right? We turn in one molecule. From the epinephrine receptor, the purple one, to the G protein, if you activate the receptor one time, it normally gives you about 100 G protein activations. Right? So there will be the first step when we amplify this. So from here to here, we've only amplified the signal 100 times. Right. One molecule will give us about 100 of these. The next step is the G protein, right? The red one moves over, it's the adenine cyclase, that's a 1-to-1 ratio. Once you activate this adenine cyclase, it's going to be doing this conversion of ATP to cyclic AMP. This one is, I think it's in the hundreds again. So one more time, going from here to the cyclic AMP, you multiply the signal more than 100 times. After that, protein kinase A, you can see you're using 4. You're using 4 molecules to activate 2 molecules. So here you basically divide the signal by 2, right? Because you're using multiple cyclic AMPs to produce 2 of these kinase. After that, that'll give us one more chance, which will be here, going from when you have this kinase activated, it's going to jump us over to hitting the APK, right? Here, I can't remember the number, but another multiplication of it, right? From then on, then it's a 1x multiplication. Because at that point, it's here, you're catalyzing, adding phosphates all the time. Here, you're transferring phosphates to the other one, right? And here, you're transferring phosphates to the sugar. So it's a 1-to-1 thing. But super powerful. You need all the molecules to be present. They can really bump up the signal.
So does this pathway always have one effect, or can it do like, is this an example and there's like multiple effects that it has on the cell?
There's multiple effects on the cell, yeah. So here it'll be like the G proteins, those things will signal on so many different levels. Before, it was like the canonical thing, or the thing that was like, oh, it only works one way, if they only thought that the alpha part did the signal. Later, a group came up and said that the beta and gamma also have their own pathway, and everyone was like, you guys are crazy with that. It doesn't happen. But then they actually showed how it has extra signals. It even has a negative feedback loop to help turn itself on. So this is like the simple version, but once you have these rules, like if you pick up, I think even protein kinase A, you'll find a library of things that it can hit. And in these kinases, they have like specific partners, but again, it's going to be those branching things that we saw earlier. You know. All right, feeling pretty good about this one. This is going to be one of my favorites, guys. Putting the, I think working on this drawing on your own, it kind of showing me what's going to be the off conformation and the on conformation. I think you guys should do it like on practice on your own. I'm gonna share, like I said, my notes with you guys today. If you guys give them back to me and you guys make your version of them or update them, that'll be the points for credit for today. And I'll give you like maybe 4 or 6 points. So go pretty well, so we can get some good points on them. Yeah.
What do we have to do?
I'm gonna give you my notes. So this is my rough notes. It's my off state on state, or you can kind of use basically off of this one, it looks a little ugly, like I'm not happy with it. But I'll give it to you guys, make it better. It doesn't have a written section. If you write it up yourself, I'll like it a lot more. My drawing skills are, so if you make it look prettier, also share it, yeah.
Is that due like 9 o'clock?
I'll take it by next Monday. I think we're gonna have a little bit more when we go through the G protein stuff next week or on Friday, so it'll make it look a little nicer, yeah. So Monday, I'll send an announcement today that we'll go through that. Question over here.
Do you have the other on-off drawings, or do you only have it for the G protein?
The G protein, for sure, you're gonna have to know how to draw that one on and off. When you do it here, you can keep it simple. If I ask just a G protein one, you have to give me a little bit more specifics. You're gonna for sure bring up the gaps in the test, yeah.
I was just wondering, like with your notes, do you have the other 3 types or no?
What do you mean,
There's a lichen-gated, the like perma one.
so that one, I'm not saying don't study it, but technically I already picked on you guys on this one pretty bad, right? So it's like you guys already had a big question for it, so I'm gonna bring it up just in the sense of like, how does it equate to one of those? I won't make you draw this one, right? This one, to me, it's really simple, right? Because only one on and off switch is being modified. So I probably wouldn't make you, like you can read it over. You'll see that it's gonna be the shortest drawing you do, but I'm not gonna push too hard on it. This was kind of easy. And the G protein is the one we're talking about right now. So yeah, G protein will be the big guy. All right, any more questions? Kind of brought that up throughout my talk. Ignore this one. Oh, sorry, so the book where I learned this from, they drew it like this, so it looks a little bit different. They have a little bit more characters to how it works. I don't know if, so it doesn't help you guys, and to put it in my notes, if you want to use it for inspiration too, draw the different pathways, it's almost the same thing, right? That green thing, we're talking about the receptor itself. The red thing would be the signaling side. This beautiful thing right here is going to be the Devlin side case, right? That's basically this guy over here. And then this is how we're basically moving the symbol, right? ATP, ATP, and there it is. So Google some, find some creative stuff, you know, have fun with it. Again, I'll share mine, it'll look great, yes, to make it better, and for sure mine doesn't have a written portion. And I really want you guys to add that to yours. So the quiz question the midterm question I'm gonna put on this one, I want you to think about, keep track of the ones you can find about how the signal gets multiplied is one. And the other one is, the really, really important thing about this type of communication is that at every place when you're transferring signal from one molecule to another, it's being done with precise interactions, right? The amino acids you have here are set up to interact with this thing to help it give that GDP to it. This molecule has a good interaction with the Devlin cyclase to make it specific so that it activates the pathway, right? It has specific pockets that help it catalyze this reaction. Same thing here. The receptors, your repressor domains, they take cyclic AMP, have that nice precise interaction. They don't grab onto the ATP. It only does the cyclic AMP, right? And then again, one more time. This is a kinase. It knows how to go and find a specific target and phosphorate it. So in my head, I keep thinking it's that handshake thing, right? Different types of handshakes, and they all have specific handshakes with each other, how they can recognize and transfer signal.
So whenever you share this to us, is like the entire class gonna be working to like edit it together, or like is each of us gonna have a bit of it to edit on our own?
On your own, the whole thing on your own, and be creative. However, it helps you memorize it. Like I said, I know some people that they can literally get flashcards and then memorize it that way. Some people, they're better at like writing in the story of what's happening. And I'm a picture guy. Like I look at this and I'm like, I'm ready to go. if I ask the question about like, what would happen if I break the interaction between like the admin side phase and the alpha subunit, I can try to figure out what would happen, right, off my chart. I'd be like, oh, the signal could come in, this will get turned on, this could be split, I'll have all the alpha GTP I want, but it's never gonna get in the side phase, the signal's basically out. Right? Any questions? All right, cool, that's good. See you guys on Friday.
You need to be prepared for quiz time. I get it.